Okay, welcome back. We are talking about how transcription occurs. Transcription, if you remember, is the process of DNA being used to create an RNA template, or DNA used as a template to create RNA. So transcription is the first stage of gene expression, and the RNA strand is going to be complementary to the template DNA strand, and it is going to be similar to the coding strand. So the first step is initiation. And for initiation, RNA polymerase is going to bind to a sequence of DNA called the promoter. So when you have the gene, right before the gene is going to always be a promoter. And that promoter is what allows that gene to be transcribed. It's not part of the it's not going to end up as part of the RNA or part of the protein sequence, but it allows the RNA polymerase to bind. When the RNA polymerase binds, it's able to unwind the DNA to separate those two template strands, to separate those two strands. And instead of copying both strands, it's only going to copy one strand. So if this is a template strand, it's just going to start base pairing and come along. It's going to keep coming along, copying RNA and making RNA that base pairs with the DNA. And eventually it's going to get to a termination site. And at that point, the RNA polymerase is going to let go of the DNA, let go of the RNA transcript, and that RNA transcript is now ready to go and be processed. RNA synthesis occurs with the enzyme RNA polymerase. RNA polymerase does not need a primer, so we don't have any primer going on or any replacing primers. You just jump right onto the promoter sequence and start making RNA. And it only goes until it hits the end of that gene, until it hits that signal that says you're done. RNA synthesis follows the same base pairing rules as DNA did, except uracil substitutes for thymine. So when there's an adenine on the transcript site, the transcript strand, you're going to end up putting in uracil as its base pair. DNA sequence where RNA polymerase binds is called the promoter. In bacteria, when it finishes the whole transcription of that gene, it ends at a place called the terminator. The terminator terminates, it stops transcription. And this piece of DNA is called a transcription unit. So a transcription unit, because it is all works together to create the transcription, to create that RNA transcript. There are three stages: initiation, where RNA polymerase binds; elongation, where it is going along, following along DNA base pairing and elongating your RNA transcript, making it longer, adding RNA; and then termination, where it hits that termination sequence and lets go. When RNA polymerase releases, you end up with a completed RNA transcript. Promoters signal the transcription start point, and they usually extend a couple dozen nucleotide pairs upstream. So you're 24 or maybe 50 nucleotides upstream. There's also transcription factors. Transcription factors help RNA polymerase to bind and help start that transcription process. And so you end up with this whole complex of transcription factors on RNA polymerase that are bound to the promoter, and that is called the transcription initiation complex. So it's not only the RNA polymerase, but also all of these transcription factors also bound to the DNA that create that transcription initiation complex. And they all work together to make it start. RNA polymerase typically isn't just going to bind anywhere by itself. There's also a promoter called a TATA box, and that is a box where you just have T's and A's and T's and A's, and that's why it's called TATA, T-A-T-A, T-A, T-A, T-A. And that really helps form the initiation complex in eukaryotes. As RNA polymerase moves along DNA, it untwists the helix. It works 10 to 20 bases at a time, so it untwists them to pull them apart to allow those bases to pair in. So you can see here's a T, so it's going to add an adenine to the transcript. Here there's an A, it's going to add a uracil to the transcript. Here's an A, so what is it going to add next? Okay, so it's going to add a uracil next, and then a C, what is it going to add next? It'll add a G after that. Transcription progresses about 40 nucleotides per second in eukaryotes. That's really fast. That's faster than I can think. A gene can be transcribed simultaneously by several RNA polymerases. So if we go back here, you could have an RNA polymerase start and come along, and as soon as it's moved over here, you could have another RNA polymerase start and move along. When it comes off, it could come right back. So you could have several RNA polymerases at the same time reading that, so you can make a lot of RNA really fast. And nucleotides are always added to the 3' end. They always move that 5' to 3' direction. When we're talking about bacteria or eukaryotes, we're going to have a slightly different version of termination. In bacteria, the polymerase stops transcription at the terminator, and the RNA can be translated just as is. In eukaryotes, RNA polymerase will transcribe a polyadenylation signal sequence that goes about 10 to 35 nucleotides. Then it moves about 10 to 35 nucleotides past that sequence when it lets go of everything. And then that RNA is going to be processed in a eukaryotic cell. So enzymes in the eukaryotic nucleus will modify that. They will go through RNA processing, and it has to go through the RNA processing before it's able to move to the cytoplasm and control translation. So during RNA processing, both ends are going to be altered. One end is going to have a 5' cap, which is a modified guanine nucleotide, and a poly A tail at the 3' end, which is a bunch of A's, a bunch of adenosines. 50 to 250 adenine nucleotides are added at the end. Then usually we also are going to cut out certain pieces of this RNA. So here's our RNA. Here's our 5' untranslated region. Here's our 3' untranslated region, that polyadenylation signal we just talked about. Our 5' cap, and our poly A tail, so lots of A's. But then here is the protein coding segments right here. And it's kind of in these different sections. Let's skip ahead. These different sections are introns and exons. Exons are the things that are going to be expressed. They are what need to exit the nucleus. They are expressed. And they are what's going to be translated into amino acid sequences. Introns are intervening. They're in the way. They need to stay in the nucleus. So the introns are going to be cut out. Introns can be short. Introns can be long. And then those exons are going to be attached back together. That's called RNA splicing. You take out the introns and just put those exons together. One thing that's really nice about RNA processing is that in different situations, your cell can trigger different parts of that gene to be introns or exons. So in that one gene, you could have slightly different versions of that gene expressed by replacing different introns or exons. So in one situation, part of this might be an exon or this might be an intron. You can change that and end up with different proteins to really have exactly the situation that you need, really be able to control the situation that you have. Okay, let me go back. It's another. There's some different reasons we have these modifications. Introns and exons can create different coding sequences to have different proteins and variety and be able to adapt to different situations. But these, especially the 5' cap and poly A tail, seem to help export RNA to the cytoplasm. So it helps get the RNA out of the nucleus into the cytoplasm so it can start working. They protect RNA from hydrolytic enzymes. So there are enzymes that are going to just be chopping up RNA as they get to it so that you don't end up with a lot of old RNA just filling up your cell. And so by having all of this poly A tail here, you're chopping off pieces of the tail, so you still have this functional protein while you chop off the tail. They also help ribosomes attach to the 5' end. So by having this 5' cap, it helps the ribosome attach so that you can start making the protein. Okay, we just talked about splicing. Be able to tell me what an intron or an exon is, what is expressed, what is not. In some cases, RNA splicing happens at a spliceosome. Spliceosomes are going to be different proteins and RNAs together, forming this spliceosome complex, and they're going to recognize the different splice sites on the messenger RNA and just cut them out. So here's the intron getting cut out. It's going to stay in the nucleus. Here are the exons that are going to be expressed. They're going to exit the nucleus, go into the cytoplasm to be expressed. There's also ribozymes. Ribozymes are catalytic RNA molecule, so they act like enzymes. So they're RNA, that's what the ribo means, but they act like enzymes. That's what the zyme part means. They can splice RNA. And so this is kind of a little bit of a disruptive discovery, because that means that not all biological catalysts were proteins. that ribozymes can act as proteins, and also helps fill in a lot of the gaps in how the first life came to be on Earth. Okay, there's 3 properties of RNA that let it function as an enzyme. One is it can form a 3-dimensional structure because it can base pair with itself. One is that some bases in RNA have functional groups that might participate in catalysis. And one is that RNA can hydrogen bond with other nucleic acid molecules. So that all makes it really good at forming a spliceosome and splicing RNA. Some introns have sequences that could regulate gene expression. So even if this intron isn't going to be expressed, it could still help regulate that gene expression. Some genes can encode more than one kind of polypeptide, depending on which sections are treated as exons or as introns. So that is called alternative RNA splicing, that you can splice this one gene in different ways. So instead of having 3 exons, you might keep this intron and turn it into an exon. You might turn this intron into an exon. You might turn an exon into an intron. You can get very different genes. So the number of potential proteins that an organism can produce is in fact greater than its number of genes, because this one can create how many different versions of this gene that do slightly different functions all off of one gene just by splicing it differently. Proteins often have molecular architecture that is different regions called domains. So there's this region that's a domain, this region is a domain, this region is a domain. And often different exons are going to be what encodes for different domains. And so exon shuffling can also result in evolution of new proteins. We have new domains being expressed that suddenly are able to bind to new things or interact differently and create different reactions. Genetic information flows from RNA to protein through the process of translation. A cell translates RNA into a protein with the help of transfer RNA. Transfer RNA are these little guys that kind of look like inhalers to me, and they have an amino acid attached to them. They have an amino acid at one end, and on the other side, they have 3 RNA nucleotides. Well, the whole thing is made of nucleotides, but these specific 3 RNA nucleotides are really important because these can base pair with the messenger RNA. So tRNAs are going to bring amino acids to the growing polypeptide by base pairing with the RNA, and it takes this ribosome to hold it all together. Translation is going to be a little bit complex, so we're going to go through it. Both in terms of its biochemistry and just the mechanics of it can be a little complex, but it's also really interesting. So we're going to talk about how we now are making proteins. Every tRNA molecule can enable translation of an mRNA codon into a specific amino acid. So it has a specific amino acid on one end, and it's going to have a specific anticodon at the other end. That anticodon is 3 nucleotides that can base pair with a complementary codon on RNA. So this AAG will base pair where you have a UUC, and it will always bring in this amino acid. Or it'll always bring in this amino acid, whatever is attached here. tRNA molecules are typically about 80 nucleotides long. They are a type of RNA. They are made of RNA. If you flatten it out, it would make this kind of T shape, which is kind of cool. Although the T actually stands for transfer and not for the shape. But it forms this shape if you flattened it, or it kind of curls up to this little inhaler shape. So the tRNA can twist and fold into this 3-dimensional molecule and forms this roughly L shape, 3-dimensional shape. Here it protrudes. That's where an amino acid can attach, and it always is going to carry the same type of amino acid. Translation requires 2 steps. First, the tRNA and amino acid have to be correctly matched. So this, has the anticodon AUA. It can only attach into one specific amino acid tRNA synthetase. And those amino acid tRNA synthetase, this one fits into tyrosyl tRNA synthetase. So that will make tyrosine be stuck onto it. So tyrosine and this specific amino acid are all that fit into this, just like a lock and key. And then you have to have the codon and the anticodon match up. So first we are putting together the tRNA and the amino acid. And there are different amino acid tRNA synthetases for every one of the amino acids. that make the right amino acid attached to each specific tRNA. There is flexible pairing at the third base of the codon. This is called wobble, and that's how we end up with multiple tRNAs that bind to the same amino acid, where you have that little bit of flexibility in translation. Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons in protein synthesis. There are two ribosomal subunits, a larger subunit and a smaller subunit, and they are both made of proteins and RNA. This type of RNA that makes the ribosomes is called rRNA, or ribosomal RNA. So we've talked about 3 different types of RNAs. Bacterial and eukaryotic chromosomes are bacterial and eukaryotic ribosomes are a little bit similar, but they also have some very important differences between them. And so there's some antibiotic drugs that can specifically target those differences in a bacterial ribosome to kill a bacteria, so that you can kill off a bacterial infection without hurting your own ribosomes, so that you shouldn't be affected by some of those ribosomal drugs that target a bacterial ribosome. A ribosome has 3 binding sites for tRNA. The P site carries a tRNA that carries the growing polypeptide chain. The A site is the next one that's going to be incorporated, and the E site is the exit site. That's where it's ready to just go off. It's finished. So you put together your RNA, your ribosome, you bring in your mRNA, the amino acid that's about to be added comes in base pairs into the A site. It can click down, the whole thing just moves down, and then that tRNA is going to be in the P site. In the P site, it can give its amino acid to the growing polypeptide chain, so it moves this in, and then it moves down into the E site where it goes off. So there's 3 stages of translation: initiation, elongation, and termination. They all require specific protein factors that are going to aid in translation, and energy is going to be required in some steps. And let's actually pause here, where we'll come back and talk again in our part 3 video, where we finish talking about translation and just finish up everything for this semester. I'll see you in a little bit.